That time I will share my screen. Yeah, so welcome to week 6. So I hope you guys have listened to all the 5 lectures in week 6. So anyone remember what we have learned till last week? Recap. What all system we have now checked? What was the first system that we have learned? Organ system I am asking about. You can type also, okay, if you're not comfortable in speaking. So first week we have learned integumentary system. There we have learned about hair, nail, and skin. Then we have passed to the skeletal system. There we have seen about bones and how the bones are formed. Then about muscles, then how the muscles are formed. Then followed by how the muscles, what are the different types of muscle, how the muscles, especially the how skeletal muscles are able to perform their function. Then we get to know, okay, skeletal muscle coordinated with neural tissue, all these neuronal tissue and skeletal tissue along with endocrinal cells comes under the class of excitable cell, and how the excitable cell is excitable and they are different from non-excitable cell. So we get to see excitable cells are those cells which can produce action potential, they can generate action potential. In that class, then we have learned how the neural system is able to make action potential, the structure of neuron, then inside the neuron, what are the different components. Then we also get to see there is a synapse junction, and there are a lot of neurotransmitters present there, and there are two types of neurotransmitters. It can be either excitatory and inhibitory. And excitatory is generally excites the neuron, and inhibitory will do the opposite function. So all till this we have seen in the, like I was telling about a recap of what all we have learned till now. So also one more thing, this is like a total 12-week session, and already we are in the half session, right? Today is this is the week 6. So we have almost reached the half of the session. I hope you ask the motto of the course. You guys get to know more about your own body. What are the different? So whenever you go to next time to a doctor, now you know why we are taking this medicine. We can question, and also we know, okay, this is what we are doing this, right? Yeah. So, um, as last week's continuation, this week also. Okay. One important thing is not only synapse and neurotransmitters, we have also learned how the action potential is generated. So, depolarization, repolarization, hyperpolarization, what are the different ion channels, and what is responsible for each of these phenomena, and how they are transmitting across all these things we have learned. Okay. So, now coming to the next part. Um, so in the first lecture, actually, okay, sorry. Again, I forgot to give my self introduction. I think everyone was there in the last lectures also, like last classes also, live interactive sessions also. But again, I will just, if there is anyone, I will give myself introduction again. My name is Harsha. So I am a third year PhD scholar in Department of Biological Science in ISER Berambur. And I am working in ribonucleoprotein biology lab here under the guidance of Dr. R. Selvi Bharadavik. Okay. So I am also one of the TA to the course Animal Physiology, which is taken by Professor Mainakdas. Okay. So that about myself. Coming to the class. So, as every time we will go through the recap of each summary of the classes, then we will try to solve some questions. Okay. So, in the first lecture, we have learned the spatial temporal summation of signal in mesh neuron. So, Sir started by telling about the propagation of signals in a neuron mesh. In each neuron, there are like multiple neurons connected to each neuron, right? But it can be some neurons can be excitatory, some neurons can be inhibitory. So, the total effective signal transmission will be combined. It's like a mathematical formula. If one is this much positive and other is this much negative, so the net will be passing will be that positive minus negative. That much will be passing to the next one. So, sir recapped, gave a recap about that thing, which he already discussed in earlier class also. Then we also get to see there is inhibitor signal and there are exhibitor signal, as I said now. Then, the main question is interesting part is uh, what is how they is interest inhibitor signal or exhibitor signal. It's depending on the neurotransmitter, right? If it's a exciting neurotransmitter, then it's exhibit the signal. Uh, and if it's an inhibitory neurotransmitter, it can inhibitory signal. And there is also interesting one question in the activity question. The question was, is same neurotransmitter can be both excitatory and inhibitory? Did anyone know? That's the answer. Is there is any the non neuron transmitter which can act as both excitatory as well as inhibitory? You can type your answer yes or no in the chat box. What is your opinion regarding that question? Should I repeat the question? So there was a activity question in that, and I felt like it's interesting. Is there any neurotransmitter that can act as both excitatory as well as inhibitory? What happened to everyone? Yes or no? How much time will it will take to write yes or no? You guys are there, right? Okay, I guess everyone decided to not to talk today. Yes, there is same neurotransmitter that can act as inhibitory as well as like excitatory. For example, dopamine is a neurotransmitter that can act as excitatory in something. Thank you, Sangeeta. She said acetylcholine. I'm not sure about acetylcholine. We'll check. Okay, but I know dopamine is can act as sometimes mainly it is inhibitory, but sometimes very rare condition it can act in excitatory way also. So if you ask why that is happening, it's depending on the receptor. So when the neurotransmitter released and in what type of receptor they are going to bind, that will determine whether it is excitatory and inhibitory. And there are same neurotransmitters that can act on bind to both of these depending on the condition. Okay. The answer is yes, there are neurotransmitters that can act as both excitatory as well as inhibitory. Then sir also gave a recap about what all we have learned till. So we have learned stretch arc, reflex circuit. Then we came to neurons. So neuron in central nervous system, there are two parts, brain and spinal cord. And in brain, again there is so that is like how we are going to follow up through next few session. Okay, brain has two part, hypothalamus and we have discussed already how the neurotransmitter normally works, but there are some disorder condition also when this neurotransmitter is sometimes they keep on releasing so that it will result in more excitation of neuron, which is a condition known as hyperexcitability. So such a disorder is epilepsy also. So that is like some disorders in neurons. So the disorders associated with specific part, for example, hypothalamus with epilepsy and Alzheimer's disease, and substantia nigra Parkinson disease. Then there is some disease associated with spinal cord also. For example, amyolateral ALS, then spinal cord injury. Like there are multiple things. Okay. So that was I was telling about the recap of the first day, and also he told about the epilepsy. So epilepsy is characterized by repeated seizures. So it's due to the reason behind that disorder is the hyperexcitability. Okay. So what happens? Suppose it everything is happens normally. Unlike that, sometimes what happen? Like the brain continuously generate the electrical impulses in an orderly pattern. Like that, as the whatever the neurotransmitter they keep on releasing, and that will result to this condition. Okay, that is known as hyperexcitability. Better example is epilepsy. Okay, so next class was mainly regarding what is hippocampus. So there are different views of uh, dorsal view, ventral view, and all. And when we see the ventral view, I guess, so we can see its name is interesting because due to the structure the name comes from. So we can see the hippocampus kind of two structures at the base. And these structures, so what is the peculiarity of this? Inside that, if we are checking, there is layer kind of structure. Each layer is known as CA1, CA2, CA3, CA4. And behind these layers, there is one more part, like below that layers. So that part is known as dentate gyrus. So dentate gyrus is very important because for a long period of time, people have believed that okay, in brain there are no stem cells. So that's why whenever there is an injury, they are like people believed this for a long period of time. Then people found out that no, that is wrong. There is still some precursor cell present in the brain inside this part. That is dentate gyrus, the blue structure. Okay, so in that blue part, you can see actually the precursor cells or stem cells. Now again, if you are checking the anatomy of neurons present inside the hippocampus, for example, CA1, CA2, CA3, CA4, and all, you can see all the neurons in kind of pyramid shape. So those neurons are known as pyramidal neurons, and so due to their shape, that neuron is called as pyramidal neuron. Okay. So that are the things that we learned in the second class. Also, the interesting story he said was so like about the surgery that happened in Canada, right? Because people that time get to know that the epilepsy or the case comes from the repeated seizure, and the repeated seizure is happening in the neurons around this hippocampus. So the in Canada, the doctors decided to okay, the patient is continuously suffering from the seizure, so in order to cut that part which produces the repeated seizure, and they have removed the hypothalamus completely. So surgery was successful. The patient was never get the repeated seizures again. But unfortunately, what happened? He lost all his memory, and that was like okay, the new way to neuroscience. How this happened? How that person lost his all memory? So we get to see that this is the hippocampus is the uh, like all the memory regulation, everything is stored there. Okay. In that context, in the third week of lecture, we get to see what is epilepsy and memory. So as I said, the story of that Canada, which is happened in very long century ago. Not long century, I think he mentioned the year also in the class. What is epilepsy? Epilepsy is happened due to this repeated seizure or hyperexcitability. So then when like this is also happened in hippocampus. When the hippocampus is removed, we get to see the memory is also getting completely wiped out. Why? Because this is the center of hippocampus is the center for all the memory, right? Then now the question is how this memory is formed. Like for example, in our childhood we have done something, still we remember that thing, right? How? So in what form it is getting stored and how the neurons every time suppose we get to see some relation with that same incident, we suddenly remember, oh okay, this happened. So all these things are related to the memory. So how all these things have happened? So there are multiple questions we can ask, and sir also mentioned that till now there is no complete answer to all these questions, right? So then in the followed by next lecture, he mentioned also about long-term potentiation, which means that suppose okay in some incident, suppose I was studying, okay, I was deeply reading something, I was deeply reading something, and I stopped reading. But I went for something else also, but still in my background, in my brain, it's still working. Okay, what is happening there? What is happening? So I have stopped doing the reading, but still it's there. So how that is known as long-term potentiation. Like we have stopped the receiving signal, but still my other part of the brain is still getting excited. So that is known as long-term potentiation. And the first person who explained this in a good way was Donald Hebb. And that's why the model is known as Donald Hebb model. So what did Donald Hebb said? He said when an axon of cell A is near enough to excite the cell B and repeatedly or persistently take part in firing it, some growth process or metabolic change take place in one or both cells such that A's efficiency as one of the cells firing B is increased. So if we remove the signal intensity from A also, the B signal intensity will get increased. That's why it is there for a long time. That is potentiation. So due to the activity of A, the B is getting potentiated, right? So such activity is known as long-term potentiation. So LTP is based on Hebbian principle. So in simple way, we can say like this: cells that fire together will wire together. Okay. Then there is one more term, long term potentiations. Opposite is long term depression, right? So that happens in case of some emotional or fear memory. For example, we have already saw a snakes and we got scared, but whenever we are seeing that same thing, okay, we are getting scared, and that kind of memory long is known as long term depression. So this is expressed by long lasting decrease in the efficiency of synaptic transmission, in particular synaptic transmission that is mediated by synaptic activation of AMPAR. These are the glutamate receptors, and it may also involve some presynaptic and postsynaptic mechanism. So just remember this long-term depression is characterized in emotional and fear memory kind of situation. Okay. So that was the recap of the class. Uh, I hope even though everyone went through all the lectures, I just wanted you to remember all the lectures so that we can go and start solving the questions. Yes, so let's go to the first question. Okay, I will read the question. You guys can start typing the answer. Which of this brain region is associated with memory and learning? Option A, hypothalamus. Option B, amygdala. Option C, visual cortex. Or option D, hippocampus. We just discussed, so I don't think there is no need of explanation. If you listen to me, I can see the correct answers here, but it's fine. Just uh, A, B, C, or D. Susri Sangeeta only answered. Okay. Shanti Priya also answered. Others, please. Four people answered. Come on, more answers. Okay, Shibani answered. Both Shibani answered. Yeah, correct answer is hippocampus, right? So we just now discussed. I don't think we need to discuss. So that has a major role in learning as well as in memory. This is the question that we have solved multiple times in the last week. But again, recap. During depolarization, which ions move inside the cell from outside? Sodium, potassium, both A and B, none of A and B. What is the ion that first enter inside the cell during the generation of action potential or depolarization? Yeah, everyone is so good in this. Everyone is so sure that sodium. Yes, very good. Correct answer is sodium. Right? Next question. This also we have solved in multiple question from the first week onwards. But again, optimum sodium ion concentration for extracellular fluid is option A 138 to 146 micromole per liter, option B 138 to 146 millimole per liter, and option C 230 to 500 micromole per liter, option D 230 to 500 millimole. Jagrati answered. Others, only Jagrati know the answer. Whatever the answer is, right or wrong, it's fine. Charumadi also replied. Yeah, very good. Correct answer is option B. So sodium in extracellular fluid is 135 to 145 millimole per liter. And in ICF or intracellular fluid, the concentration is approximately equal to 10 millimole per liter. Uh, and it's almost 35 gram around is found in the skeleton. So correct answer in the extracellular fluid, its concentration is coming in the range of 135 to 145 millimole per liter. So the closest answer is option B. So next question. Which of the following cell type form the population of the dorsal pathway of the spinal cord? This is also recap to the previous lecture. Dorsal pathway. We have learned dorsal pathway, ventral pathway, what are the different names of the same pathway, and what are the different neurons constitute in these pathways. All these we have learned. Dorsal pathway, which are the neurons you can see there. Why no one is answering? Yeah, yes, correct answer is option C, sensory neuron. Okay, just try to remember this figure, afferent pathway, which is uh, to the brain or spinal cord. So that is characterized by Afferent pathways, another name is the dorsal pathway. Okay, so afferent pathway or dorsal pathway or ascending pathway. So there are three names: afferent, ascending, dorsal pathway, which is characterized by sensory neuron. This is carrying of the information from different organs to the brain or spinal cord. And in the opposite, when it is efferent pathway or descending pathway, there we can see different types of neurons known as motor neuron, where it carries information from the central nervous system to the peripheral nervous system or to different parts of the body. AMBA is a dash. I just mentioned this point when I was telling about the long-term potentiation part. AMBA is a dash receptor. Yeah, everyone know the answer. Yes, the correct answer is glutamate receptor. So this will be helpful for you. Just remember this chart, okay? So there are two types of ion channel associated receptors and G protein coupled receptors. That is ion channeled associate is also known as ionotropic and G protein coupled is metabotropic. And in ionotropic, we can see three types of classification, which is NMDA, AMBA, kainate. Okay. And in metabotropic, there is again group 3. So all these NMDA, AMBA, all these are um, fast excitatory, and here this is also glutamine. Okay, this is also AMBA is also glutamine. You can see right GLU, GLU, GLU. So that means they are glutamate receptor. Next question is, which of the following cell types exist in the ascending pathway? Same question. Now it is ascending pathway. So you should know all the names for all these pathways. Which pathway is what? You don't get confused by different names. Yeah, so everyone is correct. It is again sensory neuron. So it is a different name for the ascending pathway or dorsal pathway, right? So exactly the same question. Which of the following cell types are part of the ventral pathway of the spinal cord? Now it is ventral, not dorsal or ascending. What constitute the ventral pathway or descending pathway? Yes, everyone is correct. Correct answer is option A, motor neuron. Okay, next question is, in epilepsy, neurons become dash, non-excitable, Schwann cell, hyperexcitable, none of them. What happens in epilepsy? Just few minutes before we have discussed. What happened? No one is answering. In epilepsy, neurons become dash. What happens in epilepsy to neurons? Yes, correct answer is option C. They definitely become hyperexcitable. Okay. Yeah, this also we have discussed at the same time. First model of learning was given by Dash. Who gave the first model of learning? If the two neurons are in close approximate position, then when the first neuron get the signal, due to that effect, the second adjacent neuron will get also fired. Or so what is who proposed that rule? Donald Hebb, Professor Meinekdas, James J. Hickman, Rosalind Franklin. Yeah, so everyone knows the correct answer is Donald Hebb. Okay, I have given the photo also in that slide if you remember. Part of the brain associated with fear is fear. That also sir mentioned the part and he also said why the name comes from, where the names comes from. The name comes from an enzyme. So if I am saying the enzyme's name, you will understand. So I am not saying the enzyme's name now. Whoever listen to the classes will answer now. Yeah, very good. Correct answer is amygdala. The name comes from the amygdalaceae. It's enzyme present in the walnuts. So due to that structural similarity to the walnuts, the name comes amygdala. Look at the structure. Okay, this one. This is amygdala. Schwann cells are. This question, if you have noted, it comes very frequently. So please be sure that if one question is regarding Schwann cell, then the another question will be regarding oligodendrocyte. So which part of the glial cells present in which part? Is it CNS or PNS? Please be careful. Don't forget, okay? Don't change. So Schwann cells are Dash cells. In which part of the brain? In which part of the nervous system? Sorry. Is it myelinating cells in central nervous system? These are myelinating cells in peripheral nervous system, or these myelinating cells in both central nervous system and peripheral nervous system. Or option D is myelinating cells specialized only for nerves in the stomach and are not found anywhere else in the body. What is the correct option? Schwann cells. We have discussed this question multiple times in previous classes also. Only Sangeeta replied. Okay, Neha replied. Others, please. Do only two people know some answer? Anything you can guess right. Yeah. So the correct answer is Schwann cells are the myelinating cells present in peripheral nervous system. So I will give a figure. So please try to remember this figure. In central nervous system, you can see these cells: astrocytes, microglia, oligodendrocytes. So oligodendrocyte is the myelinating cells around central nervous system. Oligodendrocytes, what is the peculiarity? It's very large cell. It can, one cell can myelinate multiple neurons at the same time due to its structural similarity. And these ependymal cells are also present in the central nervous system. But in the peripheral neural system, there are two types of neuroglia, which is satellite cell as well as Schwann cell. So these are present in the peripheral nervous system. Okay. So Schwann cells are present obviously in peripheral nervous system. They can myelinate neurons found in the peripheral nervous system. Okay. I'm going to next question. Now I will keep quiet. Just explain. So it's very easy, right? Tap tap answers. Oligodendrocytes are dash. Yeah, everyone is correct. So myelinating cells in the central nervous system. Yes, just now we discussed, right? Next question is an excitatory signal between two neurons. Option A increases the intensity of action potential by tenfold. Option B decreases the intensity of action potential by tenfold. Option C increases the probability of generation of action potential in postsynaptic neuron. Option D decreases the probability of generation of action potential in presynaptic neurons. Come on, everyone. I will wait for 2 minutes. Then we'll discuss. Three or four people answered already. Four people, I guess. Others, please. Yes. So let's answer now. Yes, correct answer is option C. Increases the probability of generation of action potential in postsynaptic neuron. Okay. So excitatory synapse is a synapse in which the action potential is a in a presynaptic neuron increases the probability of action potential occurring in a postsynaptic cell. So neurons form network through which nerve impulses travel, each neuron often making numerous connections with other cells of neuron. These electrical cells may be either excitatory or inhibitory. And if the total of excitatory influence exceeds that of the total of inhibitory influences, then the neuron will generate a new action potential at its axon hillock, thus transmitting the information to yet another cell. So first class. Okay, so there we have learned this. So excitatory signal can increases the probability of generation of action potential in the postsynaptic neuron. So if the question is about excitatory, then what is this is a question about inhibitory. Now you can answer very fastly, right? Come on, answer please. What an inhibitory signal between two neurons can do? Yeah, so the correct answer is option D. So just be careful. There just did presynaptic and postsynaptic neuron. So the signal always travels from presynaptic neuron to postsynaptic neuron. So the signal is always coming from the presynaptic neuron, right? So when the inhibitory signal is coming from the presynaptic neuron, that will decrease the probability of generation of action potential in the postsynaptic neuron. So option D. Okay. Now, this question also we have discussed in last week, I guess. The signal travels within the neuron is dash and between the neuron is dash. How do transmission happens between the neuron, intraneuron and interneuronal transmission? What kind of transmission can be there? Electrical, chemical, or both are electrical, or one is chemical and other is electrical, or both are chemical? What is the correct answer? Within the neuron and between the neuron. So we have already discussed that the synapse, how will they transfer, right? There is a neurotransmitter involved there. So is that chemical or electrical? If it is between the neuron, how the transmission will happen? I gave a clue also. Yeah, so the correct answer is B. Between the neuron, it is through mediated by neurotransmitter, which is a chemical. So at the synapse, it's always chemical transmission. And within the neuron or intraneuronal
B.
transfer is electrical transmission. Yes, good. So the correct answer is answer B. You will get lot of confusing options but be careful before answering. Okay, don't. Maybe you are so sure about the answer, in the excitement of the knowing answer, will mark some random answer, then maybe it's wrong. So we read the options also very carefully. Okay, so you can read this. Okay, neuron is a key player in activity of nervous system. It conveys information both electrically and chemically. That's why it's electrochemical transmission. Then within the neuron itself, information is passed through the movement of electrical charge. That charge is known as impulse, and that's why it is between within the neuron it's electrical transmission, and between the neuron it is mediated by a chemical known as neurotransmitter, which released by the synaptic vesicle will come and bind to the receptor present on the postsynaptic neuron. So that kind of transmission is known as chemical transmission. Yes. So next question. Efferent neurons, also called motor neurons, are the nerve fibers responsible for carrying signals from dash in order to initiate an action. Efferent neuron. Name itself indicates where to where. Yes, correct answer is option A, brain to the peripheral nervous system. Sorry, sorry, sorry. I answer is correct. The brain to peripheral nervous system, but option I have mistakenly typed it as B. Okay, I'll edit it. Sorry. Yes, you guys are correct. It's always brain to the peripheral nervous system. Okay, keep this figure in mind. This one or this one, right? Efferent, which is motor neurons. So it is from the brain to the effector, which is the PNS. So brain to PNS. Next question. Cell bodies of efferent neurons are in dash CNS, PNS, both CNS and PNS, none of the CNS or PNS. What is the correct answer? Cell bodies, which means the cytone, cell bodies of a neuron are present. Efferent neuron, okay? Motor neurons, they are asking about. Just now we discussed, right? Where you just need to understand where the initial point of motor neuron, starting point. Only one person answered. Sushri Sangeeta answered. Others, come on. Everyone was right about the last question, right? So, what is confusion regarding this? What is the start point of efferent neuron? Where do they originate? Now everyone started answering. One, two, three. Three people answer. Four people answer. So the correct answer is yes, option A. Since it's efferent neuron, so they are starting from the brain, so the cell bodies are present in CNS, so central nervous system. Now afferent neuron or sensory neuron. Where do their cell bodies located? Now it is afferent, okay, sensory. So they are carrying information from different parts of the body to the brain or spinal cord. So what will be the answer? Yes, correct answer is option B, PNS. Okay. Now the longest part of process of a neuron is called option A, dendrite, dendrimus, axon, aximer. Longest part. Very simple question. Very good. Correct answer is definitely axon. The conduction speed in neurons dash with effective myelination. This also we have discussed in the last class. What is the function of myelin sheath? Was the question in the last week. It's in a different way. So you know something to do with speed. So what will myelination helps to do? Simple question, right? Why so confusing? Come on, guys. Yes, correct answer is definitely it increases. So myelination helps. Myelination, the duty itself is to increase the speed of the transmission, right? So yes, it's always like insulation of wires. So it increases the conduction speed. So that was about our sixth week. We have discussed some question. So if anyone have any doubts, please feel free to ask now. Otherwise, we'll wind up the session today and I will see you on next Friday from 6 to 8. Is there any doubts? Yes or no? Is there any doubt? Please type the answer. Sure, no. If there is no doubt, then the silence I can consider it as a there is no doubt, then I will log off. I hope there is no doubt. Okay, then see you. Bye bye. See you in the next week. Bye bye. Take care. Okay, bye.